Welcome, everyone. I appreciate the opportunity to talk to you today about the work we've been doing in strawberry. I arrived at University of Davis, California, Davis in 2015 and took over the strawberry breeding and genetics program. A very long history, very famous program that's created a tremendous number of commercially important cultivars. And I came from really a background in working in hybrid crops, hybrid sunflower and some others. And so strawberry was a really new experience for me, and the octoploid genome in particular posed several unique technical challenges I'll discuss with you. The program started in 1927, and it's been a source of 73 cultivars. These cultivars have been really transformative and changed strawberry production, not only in California, but the U.S. and the world, and provide some metrics about that. So I continue to be involved in creating cultivars for the industry, and we have quite an amazing story around that. Is we have a global reach, we have international partners, and the cultivars are grown around the world, really. And so it's quite an amazing business within an academic enterprise is a way to think about it. But we work very closely with several different partners in the industry. And when I came into strawberry 5 years ago, there had really been at Davis almost no work on really the genetics or genomics of strawberry. And so our industry was really eager to have a modernization of the research program and really, as people have described it to me, to bring us into the 21st century. So that was a lot of the initiatives that I started. So just to give you an idea here, strawberry is a really strange beast. It's the result. It's only 300 years old, really, the berries you see in the market today. And it originated as hybrids between these two wild species that are both native to the New World. They were carried by explorers to Europe, and they arose as spontaneous hybrids in gardens in Western Europe. And so wild species have formed a lot of the history and background. The other thing that's unique about it is that both of the parents are octoploids. So this is really what's called a homoploid hybrid. So this was a real puzzle to untangle. We published in 2019 the first chromosome scale genome of the octoploid. I did it with my close colleague Patrick Edger at Michigan State. And then Michael Hartigan, whose paper cited here, a postdoctoral researcher in my lab, has had a really important role in not only this, but the development of the Axiom arrays that I'll be talking about in a little bit. So these diploids came together, formed a tetraploid, then a hexaploid, then this octoploid. Then we have two octoploids hybridizing to make the strawberry that we actually consume in the marketplace. So it's got a really strange history. And without the genome and the Axiom arrays and some other tools, we really couldn't unravel that history very well. So that's part of what the story I'll tell you today. The other thing that's really remarkable about this group of plants is that we see tremendous conservation of the chromosome architecture across wide genetic backgrounds, phylogenetic backgrounds, and conservation over millions of years. So very unique group of plants, all in the Rosaceae family, the rose family. So one of the very first things we had to have, and before we could start to build the Axiom array, I should back up. There was an Axiom array when we came in. It was based on a diploid, and I'll talk about that briefly. But we built a genome for the octoploid using this cultivar here called Camarosa. It's the single most important strawberry cultivar ever developed. It was grown really for about 30 years and probably on well over half of the strawberry acres in the world. So really a remarkable history and story. Was developed by a gentleman by the name of Roy Springhurst at UC Davis and his colleagues. So we got a good genome. It's got a high gene density. So that was one of the really nice features of this. You know, there's 108,000 annotated genes. but it's a 0.83 gigabase genome, so it's fairly small. And it's packed into 28 chromosomes. The basic chromosome number in the diploids is 7, so there's 4 sets of 7 chromosomes. And so this is just a plot here on the right of the gene density, and this is basically a plot of the octoploid genome against the diploid reference. Now, I don't expect you to study this eye chart in depth here, and the message I'm trying to deliver with this slide is that this is a heat map, and the red is the indicates the greatest proportion of a subgenome. So what we see in this octoploid is that these 4 diploid ancestors came together, but they had exchanges among the chromosomes. So every chromosome in the modern plant is a mosaic of the 4 diploids. It's really strange. So you think of this, you have 4 genomes come together. It's almost as if they get thrown a bit in a blender, and then they resort. Now they behave completely independently. So you've got this organism that has an octoploid history, but it's highly diploidized, so it behaves just like a Arabidopsis for all practical purposes. So this was a really powerful aspect for us, because we have diploid genetics, but octoploid genomics, if that makes sense. So one of the first things we did to start to prepare to build the Axiom arrays I'm going to talk about, was to resequence a large panel of domesticated and wild individuals. And we found very, very high levels of nucleotide diversity across the genome. That's what I'm showing in this slide. So this is one of the chromosomes here at the bottom, marked D. And you can see that there's different populations being represented, and that one of the populations has less diversity than the others. That happens to be the University of California, Davis breeding population. And we've discovered through this process that it has less diversity than the other populations. That factors in to how we design the array also, which is partly why I'm sharing that. Now, we see some chromosomes where it's not as pronounced. So this heavy line here at the bottom is a reduction of variation. But what we found in strawberry globally is that the alleles that came in from the wild, which is represented here in this Euler diagram, is that the alleles have been preserved. So the alleles came in from the wild, and they've been largely preserved through the domestication process. It's really a fascinating thing that's happened in strawberry, and there's a lot of reasons for it. So we have extensive nucleotide diversity, and this played a huge role in our ability to develop subgenome-specific assays throughout the genome. And I hope you'll find the results for the Axiom array to be interesting and exciting. Before I go there, I'll tell you what we did in parallel with this. And again, this is a bit of an eye chart with a lot of detail, but I want you to take away one idea, one message here. We whole genome shotgun sequence progeny in a giant mapping population. In fact, the parents are the 2 genomes we've sequenced. And what this allowed us to do was to understand how well we could take next generation sequencing data, so paired in 150 base pair reads, and anchor them to the genome. And so what you see here is the physical map versus the genetic map, and we get this beautiful collinearity. We were able to populate the 5,000 bins across the genome with about 1.9 million DNA variants. So plenty of DNA variants, and they're found throughout the genome. Part of what we discovered is that in domesticated populations, there have been selective sweeps on chromosomes, and there are regions of the genome that are devoid of markers. So when we started developing the Axiom array, we had this advantage of knowing that not only could we tell the 4 subgenomes apart, but we could find DNA variants throughout the genome. And what was key there is that we were able to use the wild species as the anchor, because if we had used domesticated populations, we would have been missing big stretches, haploblocks in the genome for the designing of arrays and developing the information to develop the arrays. So I've mentioned the istraw 90 SNP array, which was the first one developed for strawberry. I didn't have anything to do with it. Nala Basil and colleagues did that, and I think that array, that Axiom arrays had a tremendous impact. We used it a lot in the first 2 years of our tenure at UC Davis, and it was based on this first diploid genome that was published for woodland strawberry. And what we discovered is that array, it had tremendous utility, so don't misunderstand me, but it was, the distribution of probes was uneven, and none of the probe sequences could be anchored to our new octoploid genome. And the reason for that was they were short sequences based on this diploid, and they just didn't map unambiguously. So we went in with an entire new process. Through our whole genome shotgun sequencing, we did around 100 individuals, and that included Fragaria ananassa, which is the domesticated, and then Fragaria chiloensis and Fragaria virginiana, which are these wild parents. And on the right here, what you're seeing is the 28 chromosomes, and these are just distribution plots showing where Michael laid down probe sequences for this design process with Affymetrix. Now, how this worked was we did a bunch of in-house bioinformatics, the mapping I showed you earlier, and learning, and we were providing sequences to the team that was building the Axiom Array, and they also then applied analyses and gave us back scores. And I think that was a really great partnership, because between the two bioinformatic teams, we ended up with a really excellent design. It was a little nerve-wracking when we were doing this, because we were designing this, and of course, bioinformatically, and then sending the design off. And those were anxious moments for us. How will this go for us? So we started with the 850K SNP array, tremendously exciting thing for us, because we laid probes down every 0.1% of the genome, and just waited for the data to come back. And one of the very first things we did was use that data to look at population structure. And I'll just cut right to the chase of what happened. Out of the 850,000 SNPs, we easily got about 500,000 that were really strongly codominant. The other 400,000 or so were fine too. There were several that had no minor allele homozygotes, that type of thing. So we really focused in on that 500,000. It was way more than we really needed for most of our applications. But as the studies came together, we knew a lot about population structure, and we found that we got excellent concordance from the array to whole genome shotgun sequencing patterns of diversity. So that I don't want to go through the whole story of what's happening in these patterns of diversity. It was just really a message of saying that one concern people always have about array technology is SNP ascertainment bias. And we certainly have it. We designed the array to capture alleles that were found in the global domesticated population. But what we see is a really powerful tool to tell the story of domestication of strawberry and to navigate through that complex genome. The other thing that's been incredibly powerful for us is building genetic maps, and this is published. The arrays actually are published in this paper. We built two, the 50K and the 850K, and so we've used those in combination with some of the other approaches I talked about, to build genetic maps. And one of our discoveries was that these wild species that came together, they're completely collinear genomes. So the genes can flow freely among these. There's 8 different subspecies. They can flow freely. And what was really tremendously exciting and fun for us is that once we got the arrays and we designed the 50K array, we sent mapping populations off, and I think it was 2 weeks, 3 weeks, we had our data back. Extremely low genotyping error. You know the story of this if you've been using Axiom arrays. very low genotyping errors, highly reliable. And it was just the speed with which we could answer questions and address problems was fantastic. So that gives you really the foundation of, why we build arrays over the last 5 years. There was the istraw array. We built these two. Every probe we put on here, except for a small set from the istraw Axiom Array, is anchored to this octoploid reference. And just to let you know, we've just built phased genomes for strawberry, and we've remapped everything to those. They're really fantastic improvements, even over the first genome. And we've got the arrays re-anchored back on them. So we're still using those extensively and have some more applications that we'll be pursuing. Well, why'd we do this? Strawberry globally is in a really significant transition period. The chemical control measures that have been so important to enabling growers to reliably produce fruit have changed dramatically in the last decade, and diseases are becoming more and more problematic. The alternatives that have been available to them have been limited. So one of our most important missions the last 5 years has been to dive in to disease resistance genetics in strawberry. And this illustrates one of our very first discoveries. It actually was achieved with the istraw array, and then as we built the genome and got the new arrays, we've added on to that substantially. But we found really the first resistance gene ever discovered in strawberry, which may sound strange, was a fusarium wilt resistance gene, and we did that with Axiom arrays. To kind of segue where that's gone, we've discovered many others since then, and we've been screening diverse material from around the globe. This is a map of where all the material is coming from. Some of it's wild, some of it is domesticated, and trying to identify sources of resistance to virulent races of this pathogen that are found outside California. It's remarkable. We're in the middle of a pandemic, and one of our concerns is that we found resistance to the pathogen race that's found in California, but the concern is the way plants are moving around the world that these other virulent races will appear. And we know, sadly, from our recent experience with COVID, that this can happen in a blink of an eye. So the good news there is, again, array technology has been hugely important. This pattern of diversity we're showing here is all based on the array technology, the diversity we've sampled across the globe. And this is just a heat map showing the resistant susceptible genotypes we found. And the good news is we found more sources of resistance than we can possibly tackle in a career time. And we've done it very, very rapidly through 4 genetic approaches with array-based genotyping. Now, the arrays are fantastic, but what we need to be able to do is to apply the discovered loci in a breeding situation. So what we've done is gone in and used the arrays to pinpoint loci using genome-wide association mapping and other approaches. And this just shows that we've found the location at the top of one of the chromosomes of this resistance gene. It turns out that we found 5 other genes in that region through independent sources. And you can see the range of resistance here. This is a extremely high GWAS pink, very significant locus. It's allowed us to pinpoint, we think we have the actual gene. But we've been using Cas markers, which is a LGC technology that's widely used, to develop individual assays off of the array-validated SNPs. And we've had a tremendous amount of conversion success. I'd say well over 90% successful conversion of array to CASP assays. And we're using these now to select materials in early generations that we advance, so that we're only moving forward resistant materials. It's a very common genome-informed breeding approach and problem that is used in wheat and tomato and many other species. We're just catching up in strawberry and really bringing this technology forward. The other type of application that's really important for us is genomic prediction, and I've talked quite a lot with the team about that. But we have other diseases that turn out to be incredibly complex, like verticillium wilt. And this just shows a resistant individual, the most resistant individual we found out of a global collection, and then a susceptible individual there on the right. But this requires a completely different approach, genomic prediction or genomic selection. And again, the Axiom Array comes into play here. This just shows the distribution of probes across the genome. So what we know is that with this array technology, we have the genome covered end to end. That was really important to us. We don't want to leave any chromosome arms off or any regions, and we feel really good that we've got excellent coverage across the genome. So the general process is we form a population, which we call a training population. In this case, it was only 400 individuals. We genotype them with the Axiom Array, build a genetic relationship matrix, which is what you see here. And this is just a key saying that there's different genetic backgrounds that went into this. And we, again, sample diversity across the globe. Our goal here was really twofold. When we went into this, we had no idea that there wouldn't be gene for gene resistance as we saw for Fusarium wilt. And what we learned is that there does not appear to be classic gene for gene resistance. So we're into this complex resistance problem. I'm trying to think of some analogies here. You know, in the world of human diseases, there are a lot of human diseases that are extremely complex and for which the causative loci haven't been identified. And I think you see in the human world now, human genetics world, quite a lot of work to use genomic prediction approaches to identifying risk factors, or identifying, in our case, susceptible and resistant individuals from their DNA fingerprint as opposed to their actual disease reaction. This slide captures the essence of what we're doing there and what we've done. This paper is just coming out next week, I think. But we did this multi-year study, and what this shows is the actual observed phenotypes here on this axis. We can just look at this bottom group here, and we have 2 years of study and then the combined. So even looking at one of these plots, what these are is we use the observed phenotype and the fingerprint to get a predicted value, and you can see it shrunk down a bit. Our main goal is to get this lower end of the curve here where the resistant individuals exist. And again, with the array fingerprinting, it was very fast. We got the data back quickly. The hard part of this experiment wasn't genotyping once that technology was developed. The hard part was getting the phenotypic data and doing the statistical analyses. But what we've learned from this is that this approach works extremely well. In strawberry, we have very high reproducibility. We say high heritability, and we get excellent prediction accuracies. So those are the Fusarium and the verticillium stories are at opposite ends of the spectrum in terms of genetic complexity, but yet we've used the same genotyping technology to get answers, just simply different paths forward with that. So I'll segue in. This is really a cartoon more than anything. This is what our beautiful nurseries look like every year here in Winters, California. It's very close to Davis. We screen 12,000 to 20,000 seedlings a year, selecting for multiple traits: yield, fruit quality, disease resistance. The genome itself and the tools that the genome enabled put us in a position to apply marker-assisted selection, as we did in in the Fusarium case I showed you, or genomic selection as in the verticillium wilt case. So what we're trying to do now is translate these academic studies and proof of concept studies into action in a real breeding program. And so I said earlier in the talk that, you know, I inherited 90 years of breeding history. The university and the people in the state that are involved in agriculture want this program to continue for many years. And what we've done is overlaid now, you know, the genomic knowledge and information to help drive progress in developing better cultivars. I'm putting this out here because we don't want to continue to do this. We want to be able to select on a bench from seedling leaf samples that we quickly genotype so that what we put out in the field are all the individuals that are predicted to carry the genes that we want and need to protect this crop from diseases or to deliver the high quality or high yield. So that's the technical challenge we have in front of us. And I think the real issue for us now is that the arrays still have a significant place for us in our genetics work and many studies that are going forward. We are having difficulty applying it on the kind of scale I'm showing, an industrial scale, because we need to get to something that is much more inexpensive. The interesting thing for us is that we have cast marker technology on one end, which works for marker selection, but we need whole genome solutions, and we need a whole genome solution, a single tube solution, because when we germinate seeds in June, we have about a 4-week window before we're getting ready to transplant. So 4 to 8 weeks. So we need to get the DNA out, get the genotypes back, make this, and then perform analyses and make decisions. And that's a fairly typical problem, whether you're looking at maize breeding or soybean breeding or strawberry breeding. And so we're now in the process where I really can't talk about it today because I'm working with a couple of partners and we don't have our pilot studies totally finished yet. But we're looking at middle density solutions, 2 to 5,000 loci that we can use to apply to this kind of setting. This is something that I've had large conversations with a group at Cornell and worked with large teams that are trying to solve this same problem in many crops. And so I think there's a really important gap or hole in this sort of applied agricultural genomics area. And people are filling it with different approaches. Some people have been using genotyping by sequencing. We've used it in strawberry, it works, but it's not quite the right solution for us. So I hope that that'll inspire you and others to develop technologies for this middle density, because we really urgently need solutions that we can afford to apply on a large scale. I just need to say that I have had amazing people in my lab the last 5 years, students and postdocs, technical people, wonderful colleagues, and the support of many collaborators across the U.S. and world, and also the industry, and have had probably one of the most enjoyable times in my own career here in plant breeding, and can tell you that the Axiom arrays have played a huge role in the rapid success of a lot of our forward genetics work. So that I'll close my seminar, and thank you very much for the opportunity to talk to you today.